AP Bio. Welcome back to our Chapter 10 discussion on meiosis. So before we start, I want to kind of recover some of the ground we talked about with chromosomes. It's important to remember that an individual bar of a chromosome is called a chromatid when it is replicated, but an individual bar by itself is still a chromosome. So only a replicated chromosome has that classic X or butterfly shape, and sister chromatids, when they're replicated, should be identical. Every gene should have the exact same information. Now, every chromosome will also have a homologue or a homologous chromosome. This is a matching pair that has the same genes, but could be a little bit different. So why do we have these homologues? Well, you got one from your mom and one from your dad. So the human body has 23 pairs of chromosomes, or 46 chromosomes in total. 23 of those came from your mom, and 23 of those came from your dad when fertilization occurred. Each homologous pair has one of those from mom and one from dad. So frequently you'll see them colored differently so you can figure out which came from where, and helps you keep track. So a little bit of basic genetics. Know that when we say a gene, we're talking about a little unit of DNA that codes for a protein. A trait is what is expressed from that gene, so what we can see. And an allele is an alternate form of a gene. So this allows for the same gene to code for different traits in different people. So an example of this might be a gene for flower color. The trait is which color that is, whether it's white flowers or purple flowers. The alleles are the different forms of that gene causing those different traits. So you might have an allele for purple flowers versus an allele for white flowers. And that's going to depend on how that code in that gene is written. When we write these out, we're typically going to write them with letters. So either two capital letters, two lowercase letters, or a capital letter and a lowercase letter to show which allele the person has. And we would write two of them because you have two homologous chromosomes. The chromosome from mom will have one allele, the chromosome from dad will have a different one. Maybe those alleles are the same, maybe they're different. So that's going to come down to dominance. Now, a dominant allele will write with the letter A and is always expressed. So it doesn't matter if your two alleles on your two chromosomes are big A, or big A, little a, the dominant allele will express the trait. The recessive allele will use the lowercase letter, so little a. We only see the recessive trait if both alleles on both homologous chromosomes are recessive, so like little a, little a. Again, the dominant trait will win if they're mixed. So for example, if we mix dominant is brown eyes, recessive is blue eyes, if that child has one allele from dad that's brown, one allele from blue, the trait they will see is brown, even though they have a blue allele and a brown allele. So this is how what I mean when I say that those homologues have the same genes, but might have different information on those genes. They will have the exact same gene, but they could have different alleles. So here you can see like one, for when we have gene A, one has the dominant allele, one has the recessive. Here, both chromosomes have the dominant allele. Here we can see both chromosomes for gene D have the recessive allele. And so, two different chromosomes, but different potential outcomes. So, also, I want to remind you about somatic versus sex cells. The majority of the cells in your body are somatic cells, but then you also have sex cells, which are called gametes, and they're going to use not mitosis, which we covered last time, but meiosis. And this is going to be a specialized process that's only in gametes or only in sex cells. So only in the eggs of females or the sperm of males. And instead of making 2 identical daughter cells, meiosis is going to result in 4 cells. So it's 2 divisions, and they're going to be each one unique and different than the others. So meiosis is going to be a little bit different because it's going to have 2 divisions that result in those 4 cells. Mitosis is just producing 2 identical cells. So we're going to kind of walk through how this works. So in mitosis, you have DNA replication. Then they line up in metaphase, they split apart in anaphase, and remember, that's the chromatids breaking apart, so we get the same information in each one, and we end up with these 2 cells that are the same. In meiosis, we start the same way, DNA replication, to get those X-shaped chromosomes with the sister chromatids, but they're going to line up not in single file line, but in double file line. And our homologs are going to get separated. So now each daughter cell after that first division only has half the amount of genetic information. Those chromosomes will line up in single file line in the second division, and when they split apart, we end up with 4 unique cells because they have different different chromosomes in them, and they each have half the amount of DNA. So when we're counting chromosomes, again, a single bar is 1 chromosome. An X is a single chromosome still, it's just been replicated. So after that first split, even though there are still 46 individual chromatids in each cell, we don't have 46 chromosomes anymore. We only have 23 chromosomes. So it's no longer going to be called something called diploid. So if we break down these prefixes, you're going to see these words a lot, diploid and haploid. Diplo means double or in pairs. So the word diploid means 2 of each chromosome, your homologs, 1 from mom, 1 from dad. The prefix haplo means single. So haploid means 1 of each chromosome, or no homologous pairs. Now, I always remember this because haploid sounds like haploid. So haploid, 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 haploid. Haploid has half the amount of chromosomes because they've been separated. So during that first division in metaphase, all those chromosomes lined up double file. And when they were separated, the homologs are separated. So remember, like I said, we still have 46 chromatids in each cell here, but because there's only 23 chromosomes, those X's are still 1 chromosome, we are now haploid after the first division. Now, we don't have a second round of a DNA replication because then they're going to line up single file, and when those chromatids are pulled apart, we still only have 23 chromosomes in each haploid cell. So this means even though we divide twice and we copy once, we're still going to be haploid, haploid. And I know this is difficult to track, but it'll be a lot easier when you start mapping out chromosomes on your own with your hands. So to rehash, mitosis starts out with 46 chromosomes, it replicates, splits, and we end with 2 daughter cells, same information, 46 chromosomes, still diploid. In meiosis, we start out with our 46 chromosomes, we replicate them, but we divide from double file, so we're splitting apart those homologs. We end up with 2 cells that have 23 replicated chromosomes, and then those replicated chromosomes are going to break apart, and we end up with 23 unreplicated chromosomes in each of the 4 daughter cells that are all completely unique. So mitosis, 1 division, 2 identical daughter cells, diploid cells in somatic cells, meiosis, 2 divisions, 4 unique daughter cells. The final cells are haploid, and they only take place in gametes, or those sex cells. I know it sounds like I'm repeating myself a lot, but I really want to make sure you get these details. These are important. So you can pause here if you want to see each individual phase. I'm not going to make you memorize prophase 1, metaphase 2, anaphase 1, anaphase 1, telophase 1, all that stuff. But you can probably figure it out. If you know the steps of mitosis, prophase, metaphase, anaphase, telophase, it just happens twice. So it's not that difficult to keep track of. The big important thing to remember is in metaphase 1, those chromosomes line up in double file line, so the homologs get separated. And then in metaphase 2, they line up in single file line, so the chromatids separate. There's no additional S phase, so we don't replicate again. We only replicate at the very beginning. So in prophase 1, there's a special event that happens between homologous chromosomes called crossing over or synapsis. This is where these chromosomes exchange genetic information. So a little bit of one homolog will break off some of their DNA and trade with a little bit of another chromosome. So you can see they swap that information. So there's a little bit of the blue one on this pink one, a little bit of this pink one on the blue one. They're swapping DNA. This explains why we have 4 completely unique daughter cells and not 2 sets of identical haploids. So without crossing over, you would have one set that looked the same and another set that looked the same. So you'd have 2 different sets of unique cells. But each daughter cell is going to be unique because of crossing over, where they're able to break off some DNA and exchange it with their homolog. So here we can see the chromosomes replicate, they cross over, they divide, and then they divide again. And we end up with 4 completely unique. So again, at the beginning, we replicate, cross over, double file separate the homologs, and then break apart the chromatids. One last time, prophase 1, replicate, crossover, double file separation, single file separation, 4 unique haploid cells. And so when these chromosomes in metaphase 1 line up in double file line, the order of chromosomes is not always the same. So what that means is it's not all the moms on one side and all the dads on the other side. Sometimes they flop back and forth. So you see here, if we just had those 4 chromosomes, they could line up blue on both sides, red on both sides, or could be blue on one side with red of the other. We could have different ways of lining up those chromosomes. This is going to give us a random chance of mixing chromosomes. So this means all of our sperm cells and all of our egg cells are going to be unique. So one set of meiosis is going to be totally different than another set of egg cells or sperm cells as a result of meiosis because of independent assortment. One chromosome does not affect how another chromosome pair lines up. So if one chromosome pair is mom-dad on this way, it doesn't mean the next one's going to be the same. It could flip. It could flip all the way down. And so that's a lot of opportunity for mixing. And this principle of independent assortment is how we get these Punnett squares. Because of independent assortment, when we look at the chromosomes, we could have all these different variations of how we build a sperm or an egg based on mom and dad's chromosomes. And so that's how we get the sides of a Punnett square. We'll learn more about this a little bit later on in the unit. So there's lots of opportunities for variation among the sperm and among the eggs. So in crossing over, in independent assortment, and then finally in fertilization to make that zygote, when the sperm fuses with the egg and makes that zygote cell, whatever sperm is chosen and whatever egg is chosen produces totally different outcomes. So what are the odds of having identical non-twins? So mom and dad have the same genetic code always, but when they have siblings, they always look different. They never look exactly the same. So why is that? Well, look at independent assortment. So that is 2 to the n, which n is how many homologous pairs we have. We have 23 pairs. So that means just by mixing up how they line up in that metaphase 1, there's over 8 million different gamete combinations in one human. So unless you're about to have 8 million kids, you're not going to have 2 that are the same. But because of fertilization, you have to actually multiply those together, over 8 million times 8,388,000 again, because mom's doing independent assortment and dad's doing independent assortment. So now we're at over 70 trillion combinations just as a result of independent and fertilization. So unless you're planning on having 80 trillion kids, you're not going to have two that are the same. But even if you had 80 trillion kids, because of crossing over, you now have a near infinite amount of possibilities of what genes get switched, where they go. And so it's almost statistically impossible to have two kids who end up with the exact same DNA unless they are identical twins. So sometimes, though, meiosis isn't perfect and we can have problems. This is called non-disjunction, where chromosomes fail to separate properly. And this leads to gametes with an irregular number of chromosomes, so either too many or too few. So, for example, during the first division, during the second division, it's possible that those chromatids don't actually break. And so you end up with too many chromatids in one cell or not enough in another. Additionally, in the first division, you could have it where both of those homologous pairs go to the same cell. And so you again end up with either too many or too few chromosomes in that final product. This can result in a number of diseases you may be familiar with. So things like Down syndrome. This happens when the 21st chromosome fails to separate properly, and the child ends up with 3 chromosomes of that 21st pair. Klinefelter syndrome is a result of getting 2 X's and a Y. This is where you have someone who's phenotypically male but maybe has some female secondary sex characteristics. Turner syndrome is you only have 1 X chromosome and has a variety of different physical deformations, like some extra skin around the neck, limited sexual characteristics, and are typically very short. Jacob syndrome, XYY, too many Y chromosomes, typically ends up being very tall, usually has major acne, can have some extra expression of secondary sex characteristics. You probably have more testosterone, so there's more impulsivity. Finally, XXX syndrome. This is actually not really that big of a deal. Sometimes women are a little bit taller, but for the most part, women actually deactivate their second X chromosome. It becomes condensed into something called a bar body. And so XXX doesn't really cause any problems because it's going to be deactivated as a bar body anyway. And this bar body thing is actually why we have something called calico cats and why they're only female cats. So say a cat is heterozygous, so that means for fur color. So that means they have 1 allele for orange fur and 1 allele for black fur. Well, those cells divide in the cat, and all those fur cells are going to deactivate one of their X chromosomes. So in half of the fur cells, the orange allele gets deactivated. In half, the black gets deactivated. And because it's totally random which one it does, that's why you get this random fur pattern of black and orange fur all over the body. So meiosis can explain, and those bar bodies can explain calico cats. Pretty cool. So that's it on meiosis. Key takeaways. You should have a thorough understanding of the steps of meiosis. You don't need to memorize individual stages, although you should know prophase 1 is when that crossing over happens. And you should know metaphase 1 is how we get independent assortment because it lines up in that double file line and it's random how they organize. You should know that there's 2 rounds with 4 unique daughter cells. You should know when it's diploid and when it's haploid. So remember, the final product of mitosis is diploid. It's always diploid in mitosis. In meiosis, it's diploid at the start, but after that first division, because the homologs separated, it's haploid, and then the final product stays haploid. And then you should be able to explain how non-disjunction happens, so the chromosomes not separating properly, and that it can result in some medical conditions. Most of the time, when non-disjunction happens, the embryo won't actually develop, but there are some cases where the embryo still develops, but there is some lingering physical deformities or problems. Hopefully, this made sense to you, and I hope you have a great rest of your day.